Good evening, Samisht. Okay, so I'm Rahul Saha, and today we'll be discussing more about the C3 cycle that we had commenced in our last discussion. Okay, so in the C3 cycle, I had explained to you that how does the phosphoglycerate formation occur, okay, and how does the 3-phosphoglycerate is a 3-carbon molecule. Because of that, the C3 cycle has got its name, the C3 cycle, the Calvin cycle. Now the 3-phosphoglycerate, it goes in the reversal pathway when it comes to glycolysis. Compared to the glycolysis, the reversal of glycolysis. So we will be having 1,3-bisphosphoglycerate, then we'll be having the degradation of that 1,3-bisphosphoglycerate into, you can say, again glyceraldehyde 3-phosphate, okay, going for the glyceraldehyde 3-phosphate generation, okay, and then if you go for the glyceraldehyde 3-phosphate generation via its reduction, okay, because that will be a reductive pathway, then glyceraldehyde 3-phosphate, one of the glyceraldehyde 3-phosphates would generate dihydroxyacetone, making it, you can say, convenient for the aldolase enzyme to act upon and then cause the formation of fructose 1,6 bisphosphate. And also the antiporter which is present is known by the name as, you can say, pi dihydroxyacetone phosphate or 3-phosphoglycerate antiporter, which will suffice the role, you can say, perpetrated by the F1 F0 ATPase in the thylakoid membrane as well, in the granule thylakoid, right? Now, I'll tell you one more thing, that how is, okay, you can say, ribulose 1,5 bisphosphate, you can say, is regenerated. How is it regenerated? Because the thing is, understand it very, very carefully, that I told you one ribulose 1,5 bisphosphate will react with one carbon dioxide, okay. How will you regenerate? Okay, that is why that was the starting point. But what happens is, just listen to this carefully, 3 ribulose 1,5 bisphosphate reacts with 3 CO2. How many carbons? This has 5 carbon each. 5 3 is 15 plus 3 18. Now you have 18 carbons in total. Okay, so what you can go for is 3 ribulose 1,5 bisphosphate plus 1 CO2, 18 carbon, 18 by 3, 3 phosphoglycerate is a 3 carbon molecule. Okay, it is a 3 carbon molecule. 3 6 is 18. Okay, you can make up till 6 3 phosphoglycerate molecules if you want. Okay, now in order to regenerate this, okay, 5 of those 3 phosphoglycerates, because 5 into 3 again is 15 carbon, this is 15 carbon, okay, complete 15 carbon because it is 3, so 15 carbon. So 5 of those 3 phosphoglycerates will be used up to produce 3 ribulose 1,5 bisphosphate, complete regeneration, and one remaining, you can say, 3-phosphoglycerate, okay, 3-phosphoglycerate will be used up to go for the starch synthesis and sucrose synthesis, the C3 cycle, okay. So just understand the perspective. If the nature had used one-on-one reaction instead of going for 3 ribulose 1,5 bisphosphate, if the nature had gone for only ribulose 1,5 bisphosphate, okay, and one carbon dioxide, just like I had explained to you, okay, because I was explaining as a, you can say, as an introductory, you can say, guide in the first class, because I didn't want to introduce a 3 plus 3 concept in the beginning. So if this was true, what would have happened? 5 carbon plus 1 carbon, total 6 carbon, Now how will you be able to regenerate what you are producing is 2,3 phosphoglycerate from here, right? Because that is 6 carbon, this is 3 carbon each, they will be able to produce 2 3 phosphoglycerate. Okay, so when you are able to produce 2 3 phosphoglycerate, then how will you be able to regenerate, okay, regenerate the entire ribulose 1,5 bisphosphate, because one of them will go towards the entire C3 cycle and only 3 carbon is remaining, which is not equivalent to 5 carbon. You can never ever regenerate ribulose 1,5 bisphosphate by that method. You can never ever be able to regenerate. That's why nature plays so amazingly, so wittingly, you can say, that 3 plus 3, you're able to generate all 3, you can say, because 6 total 18 carbon, 6 3 phosphoglycerate. So out of those 5 can be taken in, 5 into 3 15, 15 carbon, done. You can regenerate, and 1 still remains 3 carbon, 15 plus 3 18, 18 here, that will go for the, you can say, C3 cycle. That is how the nature pans out, the nature processes its entire theory, you can say. It's a great way to understand what the nature is trying to do, okay, and how great the nature is. So once we talk about the, you can say, part where the regeneration comes into action, okay, regeneration comes into action. Good evening, Pratham. So what I've been teaching is how does the regeneration of, you can say, ribulose 1,5 bisphosphate is possible. So just look here, okay. So in the beginning I told you that, okay, that what you have is ribulose 1,5 bisphosphate attaches with 1 carbon dioxide, because I didn't want to impose heavy concept in the beginning only, okay. So you had 2 times 3 phosphoglycerate formed, 6 carbon, 2 into 3 6 carbon. Now by this 1 3 phosphoglycerate, if it undergoes the C3 cycle complete, reversal of glycolysis, formation of stars that we have talked about in the last class, 1 will remain, which is a 3 carbon molecule. Now how can you regenerate from 3 carbon a 5 carbon molecule? It is impossible to regenerate a 5 carbon molecule like ribulose 1,5 bisphosphate from a 3 carbon molecule like 3 phosphoglycerate. But without regeneration, the next cycle cannot happen. If ribulose 1,5 bisphosphate level gets depleted, Rubisco cannot act. Okay, with which molecule will carboxylase activity will occur? Okay, so what nature does is it plays so wittingly that it uses this pathway. It uses 3 ribulose 1,5 bisphosphate to react with 3 carbon dioxide simultaneously, okay, via Rubisco, 3 times Rubisco. So what happens is this is 15 carbon, 5 carbon each, ribulose 1,5 bisphosphate, 3 15 carbon. This is 3 CO2 means 3 carbon, okay. So 15 plus 3, you have already got 18 carbon. Now how many 3 phosphoglycerates can be produced? Each 3 phosphoglycerate is of 3 carbon only. So 18 by 3, you get, you can say, 6 3 phosphoglycerate molecules. Now once you have got 6 3 phosphoglycerate molecules, one of the 3 phosphoglycerate molecule here can go and take the C3 cycle route, okay, form the starch, and the other 5 3 phosphoglycerates, because 5 into 3 is 15 again. So 5 3 phosphoglycerate molecules will be used to regenerate ribulose 1,5 bisphosphate, and 3 were used. How many carbons? 15. 5 are remaining. 5 into 3 15. So all the 3 ribulose 1,5 bisphosphate can be, bisphosphate molecules can be regenerated. But here you have the dilemma, 3 carbon 5 carbon. Here you can see 15 carbon 15 carbon, satisfied. That is why nature goes for 3 to 3 rule. You can check 2 to 2 will not satisfy, and more than that you don't need to go because you are able to satisfy. So 3 is the best way to regenerate all the ribulose 1,5 bisphosphate molecules which have been used up. Good evening, Arya. So that is what we have been trying to do. Okay. So now how to regenerate? Okay. How does this regeneration happen? I'll just explain it to you. Okay. I'll just explain it to you. I'll just show you an image. Okay. By which I'll explain, or I'll also draw it. Okay, for you. No issues. I'll draw it for you. So glyceraldehyde 3-phosphate molecule. Okay, you have glyceraldehyde 3-phosphate. Okay, this is 3 carbon. Okay, dihydroxyacetone phosphate, that is again 3 carbon that we have. This is we're talking about regeneration of, you can say, RuBP or ribulose 1,5 bisphosphate. Okay, again 3 carbon. When they two mix up, okay, by aldolase enzyme, what they form is fructose 1,6 bisphosphate. One six bisphosphate, again 6 carbon molecule. Okay, now the real process will start. Now we have seen another reaction, fructose 1,6 bisphosphatase will come in. Okay, it will convert into fructose 6 phosphate. Okay, bisphosphatase, so fructose 6 phosphate. Okay, that is again 6 carbon molecule. Okay, now what you can do is you have a new enzyme known as transketolase, which will cause the reaction of fructose 6 phosphate with one more molecule of glyceraldehyde 3 phosphate. Okay, one more molecule of glyceraldehyde 3 phosphate. Okay, and this is 6 plus 3 total 9. So you will form two molecules. One is xylulose 5 phosphate, one is erythrose 4,4 phosphate. Okay, so you will form two molecules. 4 phosphate, then xylulose 5 phosphate. That is 5 carbon. This is 4 carbon molecule. Okay, now xylulose 5 phosphate will epimerize, will become an epimer. Okay, to become ribulose 5 phosphate. It will become an epimer, means at one of the carbons the switch will happen. OH will go this side, H will come this side. It is an epimer that I taught you in the biomolecule chapter. The enzyme will be an epimerase. It will become ribulose 5 phosphate. Okay, 5 phosphate. Now ribulose 5 phosphate, you can say, will undergo, you can say, kinase activity, kinase enzyme. Okay, so ATP, and it will become ribulose 1,5 bisphosphate. Okay, so ATP plus PI, it will become ribulose 1,5 bisphosphate. That is true. Now this side we had erythrose 4 phosphate. Okay, that was left. That will act with dihydroxyacetone phosphate, this side. Okay, this was left, this continued, this was left. So this will react with dihydroxyacetone phosphate. Okay. So this reaction is given in your ABC, you can say, reference book. Okay, that is given. This comes in your NEET examination. Okay, so dihydroxyacetone phosphate, that is again a 3-carbon molecule. Okay, so I am explaining it in detail so that you know how to answer any NEET question. Now this 4-carbon and 3-carbon will react to form a 7-carbon molecule. Two phosphates will combine to become a bisphosphate. It will make seduheptulose 1,7-bisphosphate. Seduheptulose 1,7-bisphosphates. Two phosphate molecules. Okay. Now this is again a 7-carbon molecule. Enzyme will be aldolase. Combination 7-carbon molecule. Now this 7-carbon molecule, okay, will again react with glyceraldehyde 3-phosphate molecule. I'll just write gly 6p. Okay, 3 carbon, 7 plus 3, now 10 carbon. Okay, now since this has become 10 carbon, enzyme will be transketolase, and then we will be forming again xylulose 5-phosphate. Okay, one xylulose 5-phosphate of 5 carbon, one ribose 5-phosphate. So one ribose 5-phosphate plus xylulose 5-phosphate. This is 5 carbon, this is 5 carbon, total 10 carbon, 5 carbon. Now you can check here. Wait. Xylulose 5-phosphate was here, so this can go down this route again and make one more ribulose 1,5-bisphosphate. So now two ribulose 1,5-bisphosphate will be used and 2 ATP will be used because this will take the same route as this one. Okay, so you can go for epimerase. Okay, so how many you will be producing ribose 5-phosphate, okay, or ribulose, sorry, ribulose 5-phosphate, same again, kinase ATP, ADP plus PI, that PI will be used up to produce ribulose 1,5-bisphosphate. So now two ATPs have been used up, one here, and two ribulose 1,5-bisphosphates have been resynthesized. Now this is ribose 5-phosphate remaining. Now it will go in isomerization reaction to produce ribulose, you can say, 5-phosphate. Now again ATP, ADP plus PI will become ribulose 1,5-bisphosphate, okay, by the enzyme kinase, ribulose 5-phosphate kinase. So now you can see there's a 5-carbon molecule, 5-carbon molecule, and here also it has been resynthesized. So you have been able to resynthesize 3 ribulose 1,5-bisphosphate molecules, but 3 ATPs have been used up. Okay, 3 ATPs have been completely used up. So you can say that, okay, synthesis of each triose phosphate from CO2 requires 6 NADH, NADPH and 9 ATP, because the net result of 3 turns of Calvin cycle, you are going for 3 turns, right? Not one. 3 turns, 3, because ribulose plus carbon dioxide, ribulose plus carbon dioxide, again ribulose plus carbon dioxide. So 3 ribulose, 1,5-bisphosphate plus 3 CO2. So 3 turns of Calvin cycle is the conversion of 3 molecules of CO2 okay, with one molecule of a phosphate of the triose phosphate. Okay, so you can say that the regeneration of ribulose 1,5 bisphosphate that I just showed you, so happens in this proper way, and three molecules of ribulose 1,5 bisphosphate, okay, which is a total of 15 carbons. It condenses with three carbons of, you can say, CO2 to form six molecules of 3-phosphoglycerate. Okay, that is there. Now these six molecules I told you are reduced to six molecules of glyceraldehyde 3-phosphate. Okay, and then I told you 80 NADPHs will be used because glyceraldehyde 3-phosphate, okay, will be what? When it is reduced to glyceraldehyde 3-phosphate means NADPH is used, so 6 times NADPH is used, okay. When 6 times NADPH is used in that reaction, because 1,3-bisphosphoglycerate will become glyceraldehyde 3-phosphate, so 6 means 6 times, so 6 NADPH is used up, okay. So that is there. And one more thing is, these 3 ATP are used here, okay, 3 ATP are used here, and 6 ATP are also used, okay, in that same reaction, okay, same reaction, because one molecule of glyceraldehyde 3-phosphate is the net product of the carbon assimilation pathway. Okay, if you remember that reaction that I had explained to you, okay, in the last class, okay, you can see here the 3-phosphoglycerate molecule is converted to 1,3-bisphosphoglycerate by usage of 1 ATP, okay, and 1 pi from 3 pi. So 6 times that reaction is happening. How many ATP is being used? 6 ATP, and then 1,3-bisphosphoglycerate becoming glyceraldehyde 3-phosphate. Again, it is using 6 NADPH. So 6 ATP, 6 NADPH done. And regeneration, I told you, from the ribulose 5-phosphate to 1,1 ATP kinase is being used. So 3 ATP more. So total for 3 plus 3 conversion, okay, what you have is 6 NADPH and 9 ATP are being used, because once you reach the glyceraldehyde 3-phosphate level, molecule level for all the three proper reactions, then one of the glyceraldehyde 3-phosphate molecule will diverge and 5 will be diverting to the regeneration pathway. Okay, so that is what really happens. So what you have is, for usage of 9 NADPH, sorry, 6 NADPH and 9 ATP for this 3 plus 3 reaction. Okay, that is a proper neat question. Always remember this. Okay, that is how you are able to regenerate the 3 RUBP and also go for the subsequent formation of the sucrose, okay, and the starch as well. Okay, so this is how the things happen. Now we come to, okay, something known as, you can say, the next phase of the cell of the event, that is the efflux of the phosphate group. How is the phosphate effluxed? Okay, because phosphate efflux, I had told you, happens how? Via the triose antiporter system. Okay, dihydroxy acetone in the last class, it gets effluxed. Okay, so the pi triose phosphate antiport system, it serves one more additional function. Okay, the ATP and reducing power are needed in the cytosol for a variety of synthetic and energy requiring reactions, right? Now what happens is, these requirements are met, you can say, to an as yet undetermined degree by mitochondria. Okay, but a second potential source of energy is the ATP and NADPH generated in the chloroplast stroma only. Okay, because chloroplast can also generate that. I told you they can go for the electron transport chain as well when I was teaching PS1 and PS2 during the light dependent reaction. But neither ATP nor NADPH can cross the chloroplast membrane. Okay, it is impermeable to both. Okay, now the pi triose phosphate antiport system has the indirect effect of moving ATP equivalents and reducing equivalent like NADPH from the chloroplast to the cytosol. Okay, what happens is, I'll just explain it to you. That's a very, you can say, great pathway. We'll just enjoy it. Okay, how does it come out? ATP. So suppose this is the, you can say, the membrane. Okay, so I told you that from the chloroplast membrane to the cytosol. Okay, because they cannot cross the chloroplast membrane, ATP and NADH. What happens is, once you have, okay, 3-phosphoglycerate formation, I told you 3-phosphoglycerate can also undergo or can also participate in the pi antiport system along with dihydroxyacetone phosphate. This was another 3-carbon molecule, a triose molecule or a triose phosphate molecule that can take part in the antiport system with pi. Okay, so 3-phosphoglycerate molecule. So let's name this site as the stroma, which is equal to the matrix of mitochondria. This site is the cytosol. Okay, now 3-phosphoglycerate went in. Okay, when it comes in, pi will be thrown out. Okay, pi thrown out. Fine. Now 3-phosphoglycerate molecule, ATP will be used up to convert it into 1,3-bisphosphoglycerate. Okay, ATP will be used up. Okay, 1,3-bisphosphoglycerate. Okay, that is used. Then I can say that 1,3-bisphosphoglycerate is converted to glyceraldehyde 3-phosphate by, you can say, reduction. Something will be oxidized and pi will be released again. So this is oxidized NADP plus. Okay, this becomes glyceraldehyde 3-phosphate. Three phosphate. Now next will be glyceraldehyde 3-phosphate can isomerize to become dihydroxyacetone phosphate or not. Okay, which can come out and then pi can come in again. Okay, this can come out and again pi can come in and then dihydroxy acetone phosphate will be then converted to glyceraldehyde 3-phosphate isomerization. So glyceraldehyde 3-phosphate. Then again that will go with the glycolysis route. Okay, then that will be reduced. NAD2 will be reduced to NADH. Okay, cytosol NADH plus H plus. Okay, and then 1,3-bisphosphoglycerate. this will produce, you can say, glycerate. Now that will give out, you can say, its ADP will be synthesized, ADP plus PI, arrow will be this side, 3-phosphoglycerate is synthesized. Okay, you have a kinase molecule. Now you can see ATP could cross or not. ATP got out also, NADPH got out also. Okay, you can see both ways it is happening. Just look at this reaction. Okay, so the cytosolic glyceraldehyde 3-phosphate molecule, okay, is able to, or cytosolic glyceraldehyde 3-phosphate and phosphoglycerate kinase reactions produce NADH, ATP and 3-phosphoglycerate. Okay, so then it enters the chloroplast and is reduced again to dihydroxyacetone phosphate. Okay, which completes a cycle that effectively does what? It effectively, you can say, moves ATP and reducing equivalents from chloroplast to cytosol or not. Just look at this reaction. You have this and this out. So this is the indirect way that they are thrown out. Just imagine how the nature plays a role. Okay, 3-phosphoglycerate coming in, dihydroxyacetone phosphate coming out. So this is how the nature pans out its entire great role, which will concern the movement of NADPH and ATP from the chloroplast to the cytosol, because remember, for the NEAT level, this is impermeable. Impermeable to, you can say, this thing, ATP and NADPH. Okay, but now you are getting it here. Okay, that is how it goes on. Okay, so this is the way that it continues. One more thing that I'll tell you is, now we'll go to the C4 reaction. Okay, C4 reaction. So before going on to C4 reaction, I'll show you something known as C2 reaction, which is also given in the reference book, and many of the people, they just overlook this C2 reaction. What happens is, if you remember, I had told you that Rubisco's full form is what? Ribulose 1,5-bisphosphate carboxylase oxygenase. So 3:1 ratio it has, okay, of going for its oxygenase activity as well. So when it goes for its oxygenase activity, what it will generate? Okay, when it goes for its oxygenase activity, you have ribulose 1,5-bisphosphate. Okay, plus now it will cause the reaction of O2 because it is using its oxygenase activity. Okay, Rubisco to generate it will generate 3-phosphoglycerate. It cannot generate 2 3-phosphoglycerates here because total number of carbon is 5 and this is a 3-carbon molecule. So if you need to generate 2, you need 6 carbons, but 6 carbons is not present, is not accessible. This is one O2, not CO2. So the latter product will be a 2-carbon molecule known as 2-phosphoglycolate. 2-phosphoglycolate. This is a 2-carbon molecule. 3 plus 2, 5, 5. Now this 2-phosphoglycolate is a completely futile, or you can say useless molecule for the plant. The plant would definitely try and what only try, it will convert this 2-phosphoglycolate into 3-phosphoglycerate. Okay. Now the conversion of 2-phosphoglycolate to 3-phosphoglycerate is known as the C2 cycle because it starts with the 2-carbon compound. Okay, that is why C2, it starts with the 2-carbon compound, 2-phosphoglycolate. Okay, this is a metabolically useless product. Okay, so the reaction with O2 results in no fixation of carbon, and it seems to be a net liability to the cell. Okay, so the salvaging or the salvage of the carbon from the 2-phosphoglycolate, okay, consumes significant amounts of cellular energy and also some previously fixed CO2. Okay, but the plant has got nothing to escape this. It has to go for this reaction. Okay, so that is what it does. Okay, so I'll just show you. You don't need to remember this salvage pathway. Okay, the salvage pathway of phosphoglycolate is also known as the glycolate pathway. Okay, the C2 pathway, the C2 cycle is also known as the glycolate pathway. Okay, it's a really long pathway. It involves, okay, three organelles. Okay, it involves three organelles. Number one is the chloroplast, where it starts, then it goes to the peroxisome, then mitochondria, and then returns again. Okay, I'll just show you the pathway. Okay, I just show it to you so that you just see. There is no need to know this pathway in detail. I'll just show you the pathway. Wait. Um, Now you'll be able to see the pathway in few seconds. Okay. In a few seconds it will come on your screen. Okay, so this is the pathway that you are supposed to look at. Okay, so you can see here this 2-phosphoglycolate is converted to glycolate, then transported to, from chloroplast, this is the peroxisome. Okay, becomes glyoxylate. Okay, then glycine. Okay, transamination reaction, these are not in your course, it becomes serine. Okay, serine to glycine, peroxisome, two glycine, one serine, via transamination becomes one more glycine, so glycine plus glycine, 2 glycine comes to the mitochondrion. Okay, then reduction means oxidation, glycine gets oxidized to serine. Okay, serine comes back, then serine again becomes hydroxypyruvate, it becomes again reduced. So something is oxidized, NADH is oxidized to NAD plus, then hydroxypyruvate is reduced to glycerate. Glycerate comes here, glycerate is, one ATP is again used up. Okay, so you have phosphorylation, you have, you can say, 3-phosphoglycerate. So one ATP you are using. Okay, and you can say one NADH here, one NADH, two NADH here, you are using up. Okay, in this specific reaction. Okay, so that is the cost that you have to pay. Okay, that is the cost that you have to incur in order to salvage it, but there is no other way, right? There is no other way. You have to use those things. Okay, you have to be using 2 NADH and 1 ATP in order to salvage this. You can say 2-phosphoglycolate and convert it into 3-phosphoglycerate, which will again, now it has become 3-phosphoglycerate. Now the 3 plus 3 cycle will be completed, and now you can generate 3 ribulose 1,5 bisphosphate and can go for that single 3-phosphoglycerate. Okay, after the glyceraldehyde 3-phosphate formation, of course, that will go through the C3 cycle. Okay, so this is the entire route that you take as a, you can say, plant. Okay, so next up we'll be discussing why. Okay, because again that's a, you can say, burning question. Why is, because again nobody addresses these kinds of questions. Okay, but I like to touch the very core concepts of biology. So why is rubisco rubisco? Means why does it have co, means carboxylase oxygenase activity? Why does it have such a thing? Okay, why not only carboxylase? Why did evolution start off with this kind of pathway? Okay, why did it happen like that? So what is the definitive reason behind these occurrences? So the thing is, in the biomolecule chapter, we have been able to study, or we had studied about the Km and the Vmax. Okay, so just a little bit of application of that. Now, given that the reaction with oxygen is deleterious to the organism, okay, we know that. My question is, why did the evolution of Rubisco produce an active site? Because active site is the site of the enzyme which will attach to the substrate. But the thing is, in Rubisco, I told you it is low turnover number. Okay, that's a more concentration of enzyme is needed, more substrate concentration is needed, and plus its active site cannot discriminate between, cannot discriminate properly between CO2 and O2. That is the reason. The active site of Rubisco, it cannot discriminate properly between CO2 and O2. That is was 3:1 ratio. That is a supposed ratio. Okay, that is why in every four events, one time it will catalyze the reaction, that is ribulose 1,5 bisphosphate plus oxygen to form 3-phosphoglycerate and 2-phosphoglycolate C2 cycle. Okay, why does it happen? The thing is, this evolution occurred before, like, the time immemorial, you can say, like about 2.5 billion years ago, when production of O2 by photosynthetic organisms, okay, started to raise little by little the oxygen content of the atmosphere. Okay, so about 2.5 billion years ago, it had already occurred. So that is the data available. And before this time, you can say, there were no oxygen molecules present in the atmosphere. Okay, so what happened is, there was no selective pressure for Rubisco to discriminate between CO2 and O2. Just understand it. Since there was no selective pressure to discriminate, why would you discriminate? Suppose you are already always getting what you want. So will you learn how to evade something that you don't want? Because you have never come in contact with that thing. Okay, so you wouldn't be training yourself to evade something that you don't want. Suppose you are having a boxing match. Okay, you are able to punch your opponent and he is not punching back, so you are always, you know the offense, but you don't know defense. In case he punches back, you don't know how to duck, right? That is the problem. So he is not punching back, he is never punching back, means there is no oxygen. So why would the active site make a special conformational change that it will discriminate with high efficiency the differences which pertains to CO2 and O2? There is no need for that, no requirement. So the thing is, the Km for CO2, okay, just I'll tell you the value, the Km with respect to Rubisco, okay, of Rubisco for CO2 is 9 micromolar, okay, whereas the Km for O2 is 350 micromolar. I told you that if you have Km is what? Half Vmax. Lower Km means higher affinity for the substrate. Quickly we will reach Vmax. Okay, Vmax will be reached quickly, and high Vmax you can say. Higher affinity for the substrate. Okay, so I had explained to you this graph also. Okay, this one, substrate concentration xy. Okay, then Vmax. Now so this is the, you can say, Km value. This is the substrate concentration, and this concentration means very less substrate is needed to fill up or accommodate half of the enzyme's active site. That means it is able to bind quickly. And if you have like this, okay, have like this, okay, a lot of substrate is needed. If you say a lot of substrate is needed like this, okay, you have like this, suppose like this, or if you erase this, if you have something like this, okay, a lot of substrate is needed. Okay, this means this is part 2 is worse, 1 is much better. Okay, so the, since it was reacting with CO2 properly, so its Vmax will be higher, Km will be lower. Okay, here Vmax will be lower, Km will be higher. Okay, but still some or the other cases you would be having the effect of O2, because you can see that atmosphere now contains 21% oxygen and only 0.03% CO2. So such less concentration, even this Km value sometimes fails in 1 in 4 occasions, and because of this high concentration, it will act with or react with, you can say, ribulose 1,5 bisphosphate catalyzed by the enzyme Rubisco. Okay, so that is the main reason, and the modern atmosphere is the main reason. Okay, always know it is the main reason, because it has got more than, now it is 20% O2 because of the global warming, but it is still much more than 0.03. Now it's 0.04% of carbon dioxide, again because of global warming. Okay, so always know that. One more thing is the temperature dependence of the solubilities of O2 and CO2 is such that at high temperature the ratio of O2 to CO2 in solution increases. So if you are, this part you will just will skip for now. When you will be studying the Henry's law, the solubility and the partial pressure, you can say the relationship, then you will be able to understand. So this is the reason why it has the ability to still go for catalysis of O2 with ribulose 1,5 bisphosphate, and it has not been able to take off, or you can say eliminate, eradicate, or take out that discrimination. Okay, and incur proper discrimination, or take out that inability to discriminate. Okay, so I hope you are able to comprehend this lecture. In the next lecture we will be commencing a new cycle known as the C4 cycle and the CAM cycle. Okay, so if you have had any kind of doubts or queries, you can ask me on my personal number as always to get my prompt reply. Okay, then thanks a lot. Let's meet you.